Okay, so yesterday you guys did that reading guide on incomplete dominance and things that were not exactly as clear as the Mendelian genetics, okay? So that's what we're going to talk about today. So incomplete dominance or non-dominance. All traits are not so clear-cut as dominant and recessive. When we looked at the pea plants, right, they were tall or they were short. There wasn't any in between. For example, in some flowers, such as snapdragons and 4 o'clocks, a homozygous red flower crossed with a homozygous white flower yields a heterozygous pink flower. So some genes appear to blend together instead, and this is part of the reason why there's so many eye colors, okay? Why is this not? There we go. This is known as incomplete dominance. That's really what we're going to call it. We're not going to refer to it as non-dominance, so you can just ignore that. But incomplete dominance is when no allele is dominant or recessive, and instead they blend together. Since there is no recessive allele, use only capital letters. So remember how we always did big B, little b? This time you're going to have two different letters. For example, a red flower would be big R, and a white flower would be big W, big W. So what would the pink hybrid be? Yes?
Big E, big P?
It would be a combo of the two. So it'd be big R.
Oh, big R, big W.
Exactly. Good job. So big R, big W.
Oh, don't skip it. I'm so diminished right now.
All right, so what type of offspring would be produced by two pink flowering plants? What are the genotypes of the parents? So if we have two pink plants, both genotypes of the parents would be what? Big R, big W, right? Very good. So RW and RW. Now we can set these up in a Punnett square, right? An R and a W, an R and a W. There's mom. There's dad. Okay. Now we do the drag and drop. So what's going to go in the first box? Good job. Big R, and the next one? Big RW, big W. So the first one's going to be red, right? What color is the second flower we just did? Pink. RW, so another pink one. And last but not least, WW for a white flower. Okay? So what are our genotypes? We have 1/4 RR. Now I feel like an angry pirate or something. RR. 1/2 or 2/4 are going to be big R, big W. And how many are big W, big W?
1.
Perfect. 1/4. What are our 3 phenotypes? So what is it? Remember, phenotype, the PH stands for physical. What does it physically look like?
Red, pink, and white.
Good. Red, pink, white. So in a certain plant, flower color shows non-dominance or codominance, but the stem length shows dominance. The allele for long stem is dominant over the allele for short stem. Cross a heterozygous long stem red plant with a short stemmed pink plant. Now, this, I'm not going to have you copy down. This is the dihybrid cross that I was talking about, okay? So this is when there's 2 traits. So when you have 2 different traits, and you don't have to write this down, you have 16 boxes, all right? And you come up with all the different alleles by, do you guys remember in math learning how to FOIL? First, inner, outer, last. Okay, so you just distribute the letters. So this L can go with this big R, which gives us this one. It could go with this second R, which gives us that one. This little L can go with the first big R, which gives us that set. And this little L can go with the big R, which gives us this last one. Does that make sense? So you're just like distributing out. Okay? You're not going to see very much of this at all. That's why I just wanted to bring it to your attention. That's how you get it. All right? So you'll see some of those. And the worst case scenario that it sounds like you will see on the test is if this was blank. So how would we figure out what that is? You just drag and drop, right?
O-R-I-W.
So it would be capital L, little l, big R, big W. Does that make sense?
Yeah.
Because you're going to bring this one down. So you have capital L, capital R. Bring this over, little l, big W. All right? And I'll give you a couple sample ones of those, but you're not going to have to do your own cross like this. All right? So, codominance. Codominance. In humans, there are 4 possible blood types. You can have A blood, you can have B blood, you can have AB blood, and you can have O blood. So there are 3 different alleles that determine blood type. And these 3 alleles are written as IA, IB, and I. So alleles A and B are codominant, and allele I is recessive. So in codominance, both dominant alleles, sorry, that's so hard to see on the screen. Both dominant alleles are apparent in the phenotype of the heterozygous offspring. So for example, if your mom has type A blood, your dad has type B blood, you will be AB. If your mom has A blood, your dad has O blood, O is ultimately recessive, you will just be type A. I'm type A blood. Yes, ma'am.
Isn't it if you're O blood, you can give your blood to anyone?
Yes. O is considered the universal donor because it is recessive. It doesn't have the same markings on the cell that would trigger an immune response. So if you're B blood, I couldn't give you A blood if you were in an accident because your immune system will be essentially allergic to it, which is really bad for you. You do not want that, okay? But if you get O blood, it doesn't have either of those identifiers as A or B, so anyone can get type O blood. Are we good? Yes. All right. So genotypes and phenotypes. Let's look at this. So if you have AA, you are type A. If you are A from mom and O from dad, so A and a little I, you are type A, because A is dominant over O. If you have BB blood, you are type B. How else could you have type B blood?
One B and one a BO.
Good. A BO or B and a little I. But exactly. If you have AB, you have AB blood. Since these alleles are codominant, you have both. And if you have OO, you are O. So both your parents have to be O blood type for you to be O. And I don't mean stinky. So what type of offspring might be expected if one parent has type AB blood and the other parent is heterozygous for A blood. So if they're heterozygous for A blood, they have what? What's their genotype? One parent is AB. Okay, so we know we have AB parent. What's the other parent if they're heterozygous for A? A, O, very good. A-O or A little I. All right. So what is the genotype of the first parent? We just said it was AB. The genotype of the second parent is AO. That's the only way you could be type A and heterozygous, is if you got O from one of your other parents. From one of your other parents. Like you have 6 parents, I don't know. All right, so now we do the Punnett square, right? So here's your mom, AB blood. Your dad has AO, so he's type A, but he carries an O allele. So what are the possibilities? What's going to go in the first one?
AA.
Good job. And the second one?
AB.
Good. Third box?
AO.
Good. And the fourth box?
BO.
So we can list our genotypes, right? 1 out of 4 is AA. 2 out of 4, or no, 1 out of 4 is what? A, B. 1 out of 4 is A, O. And 1 out of 4 is BO. You guys see where we get that from each of the boxes? Okay. Right? So this is that one. This is that one. That makes sense to everyone? All right. Look, I look like I coach football with that drawing. I clearly do not.
I don't know what that is.
All right, what are our phenotypes? What blood type will the first one be? A. Very good. How come it says 2/4 of them are A?
It's because one is AA and one is AO.
Atta girl. This is type A, and this is type A. What is our other blood type?
Type AB.
Type AB, very good. And our last type is going to be
Type B.
type B. So 1/4 is AB blood, and 1/4 is type B blood. So how do you guys know if you're using a capital letter and a little letter, or if you're using 2 different capital letters? So it would tell you in the word problem, if it's dominant and recessive, traditional, regular genes, dominant and recessive, you would use the big B and the little b. All right? If it is codominant or incomplete dominant, then you will use 2 different letters like A and B or R and W. All right? So that's what you're going to have to be really careful on the test, and we'll do lots of practice so that you can determine when to use a regular Punnett square with a capital letter and a little letter, and when to use a blended Punnett square where there's 2 capital letters. All right? That's going to be the tricky part. So a man and a woman have 4 children. Each child has a different blood type. What are the genotypes of the parents and the 4 children? Holy moly, right? So what are the genotypes of the 4 children? What are the 4 possible blood types? Good. So one is O. One is AB, like you guys said. One is A. And one is... Good, B. Because those are the only 4 possibilities. So what would the parents have to be? Good. What do you guys think? AO and BO, right? You know that you have to have an A and you have to have a B, and in order to see any O child, you have to have an OO, right? See, you guys are getting this. So they would be that. All right, another component of our blood is the RH factor. This is what you were asking about when you said, what about the positive and the negative part, okay? So RH factor, some people have RH positive blood and others have RH negative blood. The RH factor is determined by one gene with two alleles. The allele for RH positive is dominant over negative. So you know what, in your notes, that's probably all you really need to know about that. Since we're not going to be covering dihybrid crosses, you don't have to worry about combining these two. All right? I just wanted to let you know that's where the negative and positive comes from. So watch this. Here we go. Boom. There you go. Nope, you can just mark out that little part of your notes. All right, multiple alleles. Multiple alleles. Many genes have two or more alleles are said to have multiple alleles. This means that there are two or more alleles for the trait. Blood type was an example of multiple alleles, right? There was A, there was B, and there was O. The best example for multiple alleles involves coat color in rabbits. Coat color in rabbits is determined by a single gene that has at least 4 different alleles. That's crazy, right? These 4 alleles demonstrate a dominance hierarchy. So the 4 alleles for coat color in rabbits in order of dominance are as follows. You guys ready? So C is full color. Full color. CCH is light gray, which is called a chinchilla color. I think that one's cute. CH is albino with black extremities, or what we call Himalayan. That's what the little H stands for, is Himalayan. And last but not least, we have little c, which is albino. All white. I don't know which one the Easter Bunny is. I think it's all white. I don't know. So these alleles are listed in order of their dominance. So the most dominant color is which letter? C. The least dominant one is baby C, little c. Okay? What would be the possible genotypes for each of these rabbits? So for the full color, it could be big C, and any of the big C, little c's. Because it's going to be full color as long as it has one what? Good job, guys. As long as it has one of these big C's, it will be full color. How about the chinchilla? What must it have? CCH, and that's dominant over CH and C. So the chinchilla could be CHCH, CHH, or CHC. Does that make sense? So you're essentially like, think about it like pulling rank, okay? Who's the least in charge after that? So as long as it has one of these, then it's good. Himalayan can be HH or HC. That's it, right? And last but not least, what should albino be? You guys got it. Let me see. I think we might just stop right here. Yeah, we can do that tomorrow. All right, so go ahead and you're done with your notes.